Hello everyone, welcome to the new session of Artificial Insemination Part 2. So in this session we are going to study about symptoms of heat, semen collection method and evaluation, semen collection method, semen storage and frozen semen and storage. So these are the different topics that we are going to cover up in this particular session. So let us start this session with symptoms of heat. The various symptoms of heat are observed in cattle, that is, the animal will be excited in condition. So in this, at the time of heat, the animals are generally in excited condition. The animal will be in restless and nervousness. So sometimes they are nervous and sometimes they are restless. The animal will be below frequency. The animal will reduce the intake of food. So at this particular time, at the time of heat, the animals require less amount of heat. So the intake of feed is reduced. A peculiar movement of limbosacral region that will be observed. So this is one of the factor. The animals which are in heat will lick other animals or also smell other animals. So sometimes they lick other animals and sometimes they smell other animals. The animal will try to mount other animals. So sometimes the one animal mount to other and the animals will stand still when the other animal try to mount it. And this also observed that sometimes the animals are stand still position when the another animal is trying to mount over it. This period is known as standing heat. So this particular time, that is the time of heat, is also known as standing heat. This extends 14 to 16 hours. And the frequent maturation, that is urination, will be observed. So very frequently urination can be observed at this heat time. Clear mucus discharge will be seen from the vulva. Sometimes it will be string like. The mucus will be seen stick to the near the past of the vulva. So say there is a mucus discharge from the vulva in the case of female. Swelling of the vulva can also be seen. So the vulvas are generally swell up. 11. Congestion and the hyperemia of membrane. So hyperemia of membrane can also be observed. The tail will be raised position. So always when they are in heat condition, their tail is in raised position. Milk production will be slightly decreased because they are reduce the consumption of food. On palpation, uterus will be turgid and the cervix will be open. So at this particular time in the female, the cervix will be open. Semen collection method. Now the second topic, semen collection method and evaluation. Various methods of collection of semen have been devised from time to time. The older unsatisfactory method have gradually replaced by the new modern technique. So we are having a new modern technique nowadays to collect the semen. So let us see that. There are three common methods. Use of artificial vagina. So this is one of the method. By electrostimulation method. By massaging the ampullae of the ductus differences through the rectal wall. So the ideal method of semen collection is use of artificial vagina. So artificial vagina is the one of the ideal method for artificial insemination. Okay, for the semen collection. For the semen collection, this particular artificial vagina is the ideal one. The artificial vagina has the following parts. The heavy hard rubber to loose, open at both the ends with a nozzle for air and water in and outlet. So there are two outlets, in and out. Okay, so they are heavy hard rubber that is 2 inches loose and open at both the ends with a nozzle for air. So it is a funnel like, and the inner sleeve of the rubber or the rubber liner. The semen receiving cone or the rubber cone. So there is a part that is called as semen receiving cone or also known as rubber cone. The semen collection tube made of glass or the plastic gratitude in cc, and it is fractions correct to 0.1 cc. So this is the reading over it, that glass tube or the plastic graduate. Inserting bag before using for the semen collection. All the parts are washed thoroughly and sterilized properly and assembled as artificial vagina. The rubber liner is inserted into the hose and inverting both the ends back by folding back from either side opening and fastening with the rubber bands. So the insulating bag, that is before using for the semen collection, all the parts should be washed thoroughly and sterilized properly, so that no disease should be spread, and assembled as an artificial vagina. So all the parts are assembled as an artificial vagina. The rubber liner is inserted into the hose. So this particular part that is called as rubber liner, that part is inserted in the hose and inverted both the end back by folding back from either side of the opening. So here, now the space between the hard rubber hose and the inner rubber liner forms a watertight compartment, and the nozzle at one end of the hose can be fixed. So here you can see in the figure, that is use of artificial vagina method. Turning through the threaded nut up or down. The water jacket of the artificial vagina is filled with hot water at the temperature that is around 45 degrees centigrade or 113 degrees Fahrenheit by opening the nozzle. The graduated semen collection tube is fixed to the narrow end and the artificial vagina hose and fastened by the rubber band. So this particular side, that is the hose side, it get fastened with the help of rubber band. Okay, and the graduated semen collection tube, we will have a semen collection tube that narrow end, from the narrow end all the semen that has been collected, okay, that is poured in this graduated semen. So here you can see this artificial vagina. The inner side of the rubber liner on the anterior side of the artificial vagina is lubricated with sterile jelly to length of 3 to 4 inches. Air is blown through the nozzle into the water jacket. So air is blown. So one side, the air is blown to the nozzle into the water jacket to create pressure in it, and the same is exerted the rubber liner to stimulate the natural vagina. So air is blown to stimulate the natural vagina. The temperature of the artificial vagina is so to be checked at each collection, and it should simulate natural vagina at the mounting time. If the artificial vagina is to mount later, if it is too cold, ejaculate may not be there. If it is too cold, then there will be no ejaculation. Okay. So the temperature of the artificial vagina should be checked. Or even if the ejaculation is there, it may be contaminated with the urine and become unfit for use. So this should be checked, that ejaculation should not be contaminated with the urine. Semen collection method. The cow or the dummy is secured in the cervix crate, and the artificial vagina assembled is held at 45 angle from the direction of penis and thrust is that angle. So at this particular angle, Okay, so direction of the penis should be there, so that all the ejaculation should get enter in the artificial vagina. The artificial vagina is held with the left hand by the right-handed person, and when the bull mount the cow, the shed of the bull will be grabbed by the operator, directing the gland penis into the artificial vagina. So the bull should think that it is penetrating the vagina, but it is actually in the artificial vagina, and then the bull gives a thrust to ejaculate, and that only the time then the bull gives thrust to ejaculate, and all the ejaculation that get collected in the artificial vagina. The operator should evince care so as not to touch the exposed part of the penis. So this things should be checked by the operator or keep in mind. After the bull dismount, the artificial vagina is taken off from the penis and the air vent is open to release the pressure from the jacket. So at this particular time, when all the ejaculation is in the artificial vagina, okay, that is taken off and the air vent is open. The water from the jacket is also drained by opening the nozzle. This allows the ejaculate to flow from the cone to the semen collection tube. So this water when flow, it allows the cone to the semen collection tube. Okay, so once the semen collection has been taken, okay, so the semen collection tube is detached from the cone. Okay, now the cone is detached from the collection tube and plugged with the cotton wool and taken to the laboratory for the examination. Okay, so this collection tube should be taken away from the cone and it is plugged with the wool so that no contamination should be there, and it is immediately taken to the laboratory for the examination. The rubber cone and the semen collection tube can be protected from the external contamination or heat or higher by covering with an insulation bag with the zip. Okay, so now this particular rubber cone and the semen collection tube, okay, that should be protected from the external contamination or heat or higher by covering with an insulation bag, and there should be a zip also so that no external contamination can contaminate this tube. Semen storage. Now after the collection of the semen, there is a step that is called as semen storage. The discovery that bull semen could be successfully frozen and stored for the indefinite period has revolutionized the artificial insemination in the cattle, and this happened in 1949. British scientists discovered that addition of glycerol to the semen extender improved resistance of the sperm to freezing. So if we add some amount of glycerol, okay, so at the time of freezing the sperm do not get freeze, and improved resistance of sperm to get freeze. So this was done by a British scientist in 1949. Glycerol acts to remove water from the sperm cell prior to freezing and prevent the formation of cellular ice crystals which would damage the sperm. So if the water is present inside the sperm, then that could, at the freezing temperature, so ice crystal can be formed in the sperm and that could damage the sperm. But when the glycerol is added, all the extra water comes out from the sperm and no cellular ice crystal formed in that case. So there is no damage to the sperm in this way. There are two methods of freezing and storing semen: dry ice and alcohol, that is minus 100 degree Fahrenheit, and liquid nitrogen, that is minus 320 degree Fahrenheit. So in these two ways we can freeze and store the semen. Liquid nitrogen is preferred because there is no evidence of fertility deterioration with the age. Okay, fertility gradually decline in semen stored in dry ice alcohol. If we keep it in dry ice alcohol, the fertility of the semen declines. But if we keep it in the liquid nitrogen, so there is no evidence now till date that there is a deterioration of the fertility with the age. Frozen semen can be stored indefinitely if the proper temperature is maintained. A recent report told of a calf born from the frozen semen stored for 16 years. So the semen, the frozen semen that has been kept for 16 years, okay, a calf has born from those used semen, that stored semen. Okay, so the fresh liquid semen can be successfully stored for 1 to 4 days at 40° Fahrenheit. Semen is usually stored in glass ampoules, and other method appear promising, particularly the French straw. So this is also one of the method. Insemination method. Now after the storing, there is an insemination method. There are different method of insemination in the different species of animal. Speculum method, vaginal method, and the recto-vaginal method. 3 methods are there. The first one is the recto-vaginal method in cattle. The safe and the best method of insemination is the recto-vaginal method of insemination. So cow which is in heat is well controlled, place it in a travis, and the inseminator will get ready by wearing a plastic apron, gum boots, and gloves. So these things should be there, that is the gum boots, gloves, and the plastic apron. The semen straw after thawing, keeping the semen straw in warm water for minute to convert the freezed semen into the liquid and the sperm become motile. So the freezed semen should be taken out and kept in the warm water so that the sperm could become motile, and it is loaded in a sterilized artificial insemination gum and it is covered with the plastic sheath. The inseminator will insert the gloved left hand into the rectum after applying the soft soap or other lubricant on the glove and back ragged the animals, and the hand is further inserted and will catch hold the cervix through the rectal wall. So the inseminator will insert the gloved left hand into the rectum and the hand will reaches up to the cervix. The artificial insemination gum loaded with the semen straw is passed there through the vulva to the vagina and the cervix and observed with the hand in the rectum that the artificial insemination gum reaches the cervix. Then the semen is deposited by the injecting the gun, and after depositing the semen, the gun is removed. The empty straw and the sheath are disordered. So this is the way the recto-vaginal method of insemination is done. Spectrum method. In this method, the spectrum is placed in the vagina of the cow, which provide passage outside to the site of insemination. The insemination tube is passed through the speculum and the semen is deposited in the cervix insemination method. So in this method, what happened? The insemination tube is passed through the speculum, okay, and the semen is deposited in the cervix. So the insertion of this insemination tube is done in this method. Vaginal method. Hand is passed through the vagina and the insemination tube is guided by the hand to the site of insemination and semen is deposited there. Here there is a risk of contamination and the injury of female genitalia. So this method is avoided. Frozen semen and storage. Freezing of semen for the successful preservation of the spermatozoa for a longer period is of great importance in the livestock breeding and the farm management. So this is one of the big challenge, that we should successfully preserve the spermatozoa for longer period of time. And it is of great importance also, right? So it has made possible to make available the use of outstanding proven size for the larger number of cows covering larger area. Frozen semen shipment has become possible to the different countries in the globe, to any place connected with any services. So this is one of the method that we can take frozen semen shipment to anywhere in the continent. Nowadays, if farmer want to use an outstanding size of inheritance of high milk yield, he can go for the frozen semen service provided. His area is covered by the artificial insemination with supply of frozen semen. So if we want, we can have this type of facility in our area. Okay, so we can have those semen of the cow that are of high milk yield. At present, frozen semen is used in most of the states in India. Okay, so this is all about this session. Thank you very much.